thank you very much for agreeing to give a Build a Cell seminar, and you can get started.
Okay, thank you, Kate, for inviting me and giving me the opportunity to present. So I'm going to quickly share screen so that you don't have to see my office all the time, and see how that goes. So I think, so this is working, I guess. Yeah, yep, so
we see the slides.
Yeah, so Kate mentioned I'm going to do my own introduction, so which I prepared a slide for that, I guess, and that is my first slide, I would say. So a little bit of the background. My name is Hans-Joachim Wieden, and most of the people know me as HJ, and I've been I'm at the University of Manitoba, where I started or moved to during the pandemic about 19 months ago, and started my lab there. So I'm going to show you a slide later on. Before that, I was, and I'm now located in the Department of Microbiology. Before that, I was at the University of Lethbridge in Southern Alberta, and spent 15 years of my life in that lab, in that institution, and built some sort of synthetic biology capacity. And we had, I established the iGEM program on the high school and the collegiate level, started a Synbio makerspace that allows emerging companies to cross the pre-valley of death, like from the idea to something that they can show that it actually works before you actually hit the valley of death. So that has been quite successful. The place does training for the Alberta high school iGEM teams. So my background is, before I moved to Canada, I completed a degree in chemistry at the University of Düsseldorf in Germany, then did a PhD on ribosome function. And I have to say that during my diploma work, so my master's work, I worked on respiratory chain complexes, so large biomolecular assemblies that actually are encoded to function as machines. And that theme, you will see, drags through my whole career, because I'm enticed by biological devices and how they're function, to unearth their function and to forward engineer them. And I guess that's partially why I'm here. So after my PhD on the function of the ribosome, or the bacterial ribosome, I did a postdoc in computational biology and in structural biology. So I'm trained in cryo-EM before cryo-EM actually was hip. Wanted to say that. Now I'm at the University of Manitoba, and have restarted the lab, or like we've successfully started a lab, and work on reconstituted cell-free synthetic biology. And that's something I want to talk to you about today, because that is the intersection of where we're interested in. So you, as I mentioned earlier, so the focus of my work is in looking at biomolecular devices and their complex interaction. And the primary focus sits on, let me quickly put a pointer on that. So from the introduction, it will go slightly into the presentation, and then we're going to go from there. So the main focus of the lab is identifying the design principles of biomolecular devices. And the focus, of course, is the ribosome. We're going to talk about the ribosome a little bit later. And with the knowledge we're getting there, we are trying to develop or construct biomolecular devices de novo by exploiting what we have learned about how they are done. So one of the major achievements recently is that we were able to identify or to develop a computer-based, computational-based biosensor design platform that we have, which is proprietary and which we are in the process of commercializing, that allows us to do designer biosensors, basically, for all sorts of small molecular ligands. So understanding the design principles also allows us to identify novel antibiotic targets, or more importantly, actually identify functional aspects of emerging antimicrobial resistance, because they're usually exploiting some of the design principles that are behind the function of the cellular components. And as you can see, I said I have a background in biophysics. We're combining computational biology, structural biology, to build or analyze biological devices and to deploy them in synthetic biology applications. What we're currently main focus of our work is cell-free cell-free based biomolecular manufacturing platforms. In this regard, is it's basically the software, like the foundation to do cell-free synthetic biology, and we're going to talk about this later today. Within that framework, what I find highly interesting is our two classes of biomolecules or biomolecular components, and those are GTPases and the decision-making process that GTPases enable in a biomolecular assembly. And our focus lies in the lab on the structural dynamics of translational GTPases, and you will see how critical they are. Why? Because how critical they are in for progressions through gene expression or on translation. We're going to go into this, and we're interested in RNA-based gene expression regulators, both on small RNAs that modulate the function of gene expression elements or devices, as well as RNA-based gene expression elements that are basically involved in the actual process of gene expression. So they are part of, let's say, the mRNA, and that's what we're going to be talking about today. And I thought that's a kind of a nice topic here. And what I would like to do today is I would, based on that background, tell you a little bit of a story of a PhD student, and I like Luke Roberts, who just recently defended, and how we have tried to build a biological initiation device, learn something about biology, and improve basically cell-free or cell-free expression systems that way. So, you know, through an exciting project, through a disappointment, through something that actually is very useful. And I also have done basically, I broke this presentation basically two parts. So we're going to go through the first presentation, and there will be a stop, and there will be like question slides. And if you want to hear more about a certain project, I have the second section that we can go, or we just can go into a discussion and talk about, have questions, answers. Okay, so in a nutshell, the way we're looking at biological systems is, and I mean, I'm probably speaking to the people, preaching to the choir is what I wanted to say. Preaching to the choir about like how is the cell looked at, but we can basically, that's always a slide I'm using in intro. Synbio is like the overall deconstruction of the main elements of a cell. This is basically it's a confined environment or confined volume, accuracy volume, usually that is confined by a cell wall or cell membrane, and that basically contains a key information flow that gives information from DNA over RNA to protein, which is based on the central dogma. I just did not include the processes that involve functional RNAs, but we're all aware about these functional RNAs, and I touched on that in my previous slide already. The reason why I'm showing this is there's always processes that give rise to the elements that are important for the progression through the functional cycle, and that is, for instance, the synthesis of the precursors that give rise to the DNA, or the synthesis of the precursors that give rise to the RNA, or the synthesis of the precursors that give rise to the protein. There's some machinery involved in that. There are particular machineries and processes in place that allow the transcription process, that allow translation, which is protein synthesis, that maintain and fine-tune DNA structure and its function. The pathways we talked about, right? There's processes and machineries in place that help with the construction of the cell wall, and processes and that have to do with the structure and the function of the cell membrane. All of that involves the intricate interaction of molecules with a pre-designed basically mechanism at the molecular level. And what we're trying to do is to unearth or discover these molecular bases of the machinery as describing it by biophysical means, in order to get a blueprint that allows basically the rational design of these processes. How critical they are, and that's how I always say when I'm showing this slide, is they're all targeted. These processes are all targeted by antibiotics that in one way or the other, or small compounds that work at antibiotics, that in one way or the other interfere with the dynamic processes that underlie the molecular mechanism or the function of these biomolecular machines within the cellular context. So the protein synthesis machinery, for instance, is targeted by 50% of the commonly used antibiotics, and for that, it's a key target. And what my focus, my research, and I indicated it before, focuses on is translation. And the key component in translation, in the conversion of RNA stored information from mRNA into proteins, the key part of that machinery is is the ribosome. So these processes, so the conversion, and again, we have seen all this slide about synthetic biology again, but taking this out of the living context and being able to do gene expression ex vivo, so in cell-free environments, basically allows us to do cell-free synthetic biology, and that can range from biocomputing to the production of value-added compounds. There are fundamentally two ways how cell-free systems exist. They're fundamentally either cell lysates that you can attenuate, but they're basically by popping open a cell of a particular type, and then basically attenuating whatever is the contents of that type. Or on a completely other end of things, a fully reconstituted, purified in vitro expression system that typically is couples transcription with translation. We all know this as the pure express system, and which has made that kind of reconstituted system available. We all know that the advantages of such a system is that we do have full control about the composition. We know which components are in there, and we can actually modify and swap them in and out as we like to do so. Also, the system is modular. You can actually design certain elements around it. You can have an energy production system. You can have a ribosome decoding element. You have the tRNA amino acylation elements, and so on and so on and so on. You can actually swap them and design them in a particular way. We also are familiar with the common disadvantages of these systems. So typically the low protein yield compared to the cellular system. So which indicates that we're probably not 100% clear what actually happens in those systems, and that's where our research comes in. They're typically expensive to make, and we have been, as a community, spend a lot of effort in reducing the cost for these systems. And but I think these would benefit from a detailed understanding of the molecular process that derive to the actual functionality of this. With that being said, at the heart of those systems is the in vitro reconstituted systems is the ribosome as the protein generator per se. The ribosome, I'm just going to give you a quick introduction on of the lay of the land of the ribosome. The ribosome is a large ribonucleoprotein complex that consists of two larger subunits, a small subunit and a large subunit. In the bacterial case, it's the small subunit is referred to as the 30S ribosomal subunit, and the large subunit is referred to as the 50S ribosomal subunit. The overall arrangement, or the overall design of the ribosome, is conserved across all domains of life. The sizes and the particular details vary, but fundamentally, the ribosome has a purely RNA made up catalytic center that is responsible for the joining of the amino acids and the synthesis of the growing peptide chain, and which is located on the large ribosomal subunit. The small ribosomal subunit will bind the mRNA that contains the genetic the information to be decoded, and the decoding occurs by transitioning by binding of amino acid tRNAs in a codon-dependent manner that then ultimately into the ribosomal A site, which is indicated here as a complex of the yellow tRNA with a protein factor that's responsible for the delivery of the amino acid tRNA to the ribosome, which is elongation factor 2 in bacteria. Codon recognition occurs. The tRNA is going to be accepted, moves into the ribosome. Peptide bond transfer occurs, and then basically from the P site tRNA bound peptide site tRNA growing peptide chain onto the incoming A site bound tRNA, and then another elongation factor binds to the ribosome, moves the tRNA together with the mRNA one step further to actually have the next tRNA coming in to decode the codon, and the spent tRNA will dissociate from the exit site, from the E site of the ribosome. So that's actually straightforward, and we know everything about translation. So basically, as I indicated, the seminar has two parts, and we can go home now. But Is it so simple? No, it's actually not that simple, because the ribosome, with its relatively straightforward and conserved synthesis process, is actually embedded in a cell that has to respond to, or it has to respond to different external stresses, which then basically also can impact the function of the ribosome. So, for instance, the availability of energy, or the availability of nutrients, for instance, changes the availability of amino acid tRNAs in a certain way. So the ribosome has to be able to deal with the lack of, or the availability of certain amino acid tRNAs. The message that is the protein that is synthesized will have different responses to the actual ambient temperature. We are using different amino acids on the same ribosome. So basically the substrate constantly changes from time to time. Some of the substrates have to go into membrane, which requires different processes to allow for that. There is allow for the insertion into the membrane, or to localize the message, or the protein to the membrane. But what also is important is there's a lot of things go wrong in the cellular context, and the ribosome actually has processes to deal with these issues. Like when an mRNA is truncated, the ribosome would otherwise be stuck on the mRNA. So there needs to be processes in place that allow the ribosome to release, or to resolve this stall, and to allow the ribosome to be recycled. So all of these kind of processes not only impact the actual progression of the ribosome through its functional cycle, from synthesizing the ribosome during ribosome biogenesis, but also through the translation initiation process, translation elongation, translation terminating, the ribosome recycling, and ultimately also the degradation of the ribosome. These are all decisions that need to be made, and these processes are fundamentally involved, are fundamentally important for ribosome function. This makes them actually important potential antimicrobial targets, and also since these antimicrobials target usually dynamic processes of the ribosome, the existing mechanisms that help them to overcome such stresses or challenges in the functional cycle might also be used to overcome antimicrobial resistance. And then we can talk about later about a case where we showed that in a lab that's the case. But most importantly, and for the audience here and for me in the lab, it actually complicates the process of using cell-free or reconstituted systems for synthetic biology and bioengineering, because all of that framework is basically encoded into the ribosome, and understanding how that is there, and separating out what is actually necessary for the ribosome to function, rather what outside of a cellular system for a particular set of mRNAs, would be important to enable targeted engineering of the ribosome or the translation machinery as a biomanufacturing platform. But for being able to understand which of those regulatory processes or interactions that are built into the ribosomal structure and function is also a key feature to understand what we need to move forward to build lifelike devices up to including a synthetic cell. So what I would like to do today is I would like to focus a little bit on the complexity of the ribosome synthesis process and of the protein synthesis process using the ribosome, by having a little bit more detail on how the elongation cycle, or actually how protein synthesis actually works. So I indicated that the process in itself is conserved, like the ribosome, conserved among the domains of life. The actual protein factors that orchestrate the transition might be different, but the overall design is the same. So protein synthesis usually on the ribosome-dependent protein synthesis starts with elongation in a relatively complex and highly regulated process, where the 30S subunit, the mRNAs, and together with initiation factors and initiated tRNA come together, form a 30S initiation complex, which then brings in the 50S ribosome, assembles the translation or the elongation competent ribosome which then enters the elongation cycle, where we go through the classic textbook kind of process of synthesizing the protein, as we discussed before, until it hits a stop codon, and then the complex gets assembled, the protein gets released, and the components will be recycled. From the point of view of a reconstituted purified system, it would be kind of great if the initiation process would be less complex. Then we would have less components that we need to provide the system when we reconstitute it, and we would have a better control of initiating translation in a more reliable way. We all know, or all of us that work on these reconstituted coupled transcription translation systems know that initiation is one of the issues that needs to be overcome to get increased the protein yield here. So having a module that would take care of translation initiation and can be just popped in front of the mRNA to do all of that would be a fantastic design thing. So with that in mind, we came across such a process, and a while ago, when the crystal structure of a RNA element of the like a 5' UTR RNA element on a bacterial ribosome was resolved, which is basically something that we call an internal ribosomal entry site that works commonly in eukaryotic cells, and that's derived from a couple of viruses. So these viruses can initiate translation in the eukaryotic system without the use of any additional factors, which has been exploited quite a bit as a defined translation initiation strengths tool to study the function of proteins within the eukaryotic context. And it would be very cool if such a similar device would exist in bacteria that would allow us to do the initiation process here. So that was the starting point of the story that I want to tell today, is that we had a crystal structure of this iris element here located in the E side of the ribosome that I introduced you earlier to. We can see that there's an RNA structural element here in red that points into the decoding side, or actually into the P side of the 30S small ribosomal subunit, and it is interacting with the 50S subunit. So my graduate student, Luke Roberts, who ran across this crystal structure, was excited about this and wanted to design this element as a cross-kingdom translation initiation design element that one could use to develop genetic circuits, or like translation elements, or as a synthetic cell-free synthetic biology device that one could design, for instance, in an E. coli-based system, and then move it into a different system, and one would not have to optimize all of that, or just develop that module as a translation initiation element for bacterial cell-free synthetic biology. And so with that, I want to give you a quick run through of what we're looking here at. So these internal ribosomal entry sites are derived from they are called the IGR irises. They are derived from an intergenic region between two open reading frames, and that would allow translation initiation in front of that second ORF, avoiding the normal scanning approach that one would have to do, or a cap-based initiation process that eukaryotes usually would use. The structural, this RNA element has three signature elements: a pseudonode 1 here in blue, a pseudonode 2 here in red, and a pseudonode 3 here in green, that form a very compact three-dimensional structure, as indicated in the crystal structure here in the bottom. You will notice that that pseudonode 1 forms a tRNA-like interaction with itself, forming something that looks like an mRNA tRNA complex, which would then mimic basically the situation in the P side of the initiated ribosome, which then is followed by here in yellow with an mRNA kind of element that then basically gets right. So it folds into a triple pseudonode structure, which I indicated here. It requires no additional factors. It binds directly to the preserved ribosomal core. and for that reason skips directly into elongation, which is basically what we wanted as a design feature for that. The IGOS structure can drive translation across multi-domains of life, and this current structure, this current element is the only structured-based initiation that we are aware of, or at least at that point in time we're aware of, that drives translation of bacteria. So with that in mind, we just started characterizing. So Luke went in and designed basically a single cell translation assay that would allow us to benchmark and standardize the translation initiation strengths of those ribosomal of those iris elements. So in what Luke has done is he designed using computational approaches, basically based on the available ribosomal binding site libraries from the registry, and using the SALIS calculator, a set of ribosomal binding sites that have a defined predictable strength, and drove translation of a fluorescence reporter by them, and wanted to see if we can correlate the predicted expression strengths of the RBS with the observed expression strengths based on the reporter. One of the other important pieces that why we did this in using flow cytometry is that we wanted to know how homogeneous the population of the cells would be that respond to that element. So if there's like if there's different population, or if this is homogeneous. So that's why we opted for a single cell approach. I will not show any flow cytometry data here, but when we ever gonna get the expression strengths of something in the cell, it's done by flow cytometry, and the populations are homogeneous, and we can separate out the dead cells, kind of thing. But it will give you, but they're all measured in flow cytometry, and they're all populations. So with that in mind, Luke ran a couple of experiments and had created basically, as I said, the different variants with different predicted translation efficiencies, and you can see that our system nicely reports, or can predict reports strong, medium, and weak RBSs and their expression levels. So we're confident that our system indeed is able to measure in vivo at the single cell level the expression levels of or the expression strengths of these different standard initiation elements, which forms the foundation for looking at the different initiation strengths of the viral irises in this system here now in E. coli. So the design of our iris construct is straightforward, and then basically mimicking all the biochemistry and all the molecular biology that is known and available for these IgA irises from eukaryotic systems, where we're just doing these now basically in the bacterial system. So we are studying, we have the three different pseudonodes, which then basically should drive translation of a superfolder GFP signal behind it. We have done this. We then wanted to know, because the crystal structure is available only for one of those virus irises, we wanted to see if this is if this is specific to that one iris, or if this is a common feature of all the IgR irises. And so we tried three different ones: the Plotia stali intestine virus, the cricket paralysis virus iris, and the Israeli acute paralysis virus iris. So PSIV, CRPV, and IAPV, respectively. So we wanted to know how the first question that we asked is basically, how does iris-driven translation relate to the translation strengths of normal ribosome, like Shine-Lagrange-based translation initiation in bacteria? And because that was not clear based on the crystal structure. And as you can see here, overall, the translation initiation strength of the structural elements is at best comparable to the ribosomal, to the expression, to the expression strengths of a weak ribosomal binding site, which is so which for us first was a disappointment. Also, depending on which virus iris you're using, this can be even less than we would expect it for a weak ribosomal RBS. So which means On the one hand, it's quite a disappointing thing because it shows you that this runs as a weak translation initiation element. On the other hand, the differences between the expression levels or strengths between the different iris viruses gives us some design, or shows that there might be some design space between the different sequences to fine tune the expression levels. And it's something we were trying to look for. So with that in mind, we then started basically identify and look for the contribution of the different structural elements. So previous work in the eukaryote system has shown that when you are basically removing or breaking the pseudoknot interactions here for the different pseudoknots, you basically lose translation initiation. And so this has been the proof for the structure-based mechanism for this translation initiation. So when we want to design the iris as a translation initiation element, we need to know what can be changed in that structural element to fine-tune the translation initiation thing. So with that in mind, we basically generated the corresponding knockout variants, or like knockout refers to we are knocking out that pseudoknot, not we're knocking out a gene or anything like that. But basically, when we say knockout here, it means we're interrupting or disrupting the formation of pseudoknot 2, or pseudoknot 3 or pseudoknot 1 in that regard. There's a couple of other experiments we have done, which I can talk about later, which we're not going to talk about in the first go around, but for now, we're just basically talking about the knockouts of the standard pieces here. So if this is a structure-based initiation element, this then would lead to decrease of translation initiation strengths by knocking out the system. So that should be corresponding to the eukaryote system, and we were kind of wondering how strength the effect was. And so now, with that being said, we did this for all of the three virus irises that we used, and I want you to focus first on the CRPV iris on top. We are comparing here the wild type initiation strengths, which we have set to 0, and compared to, for instance, the pseudoknot 1 knockout here. So this is the knockout of that tRNA mRNA mimic, and as expected, for that being a critical piece for the translation, structure-based translation initiation, exploiting basically this common structural core within the ribosome, we see a strong decrease of translation initiation. So that was great, and Luke and I were pretty excited about that. However, when we are knocking out the pseudoknot, or when we're interfering with pseudoknot 2, the translation initiation strength increases a couple of times. So now then knocking out pseudoknot 3 brings down translation initiation strength a little bit, and knocking out 1 and 3 indeed shows, as expected from the eukaryote context, brings basically translation initiation down to the level of the lack of a start codon. However, combining all 3 of those knockouts basically resulted in an express initiation strength that is strong, or that's higher than for the wild type iris element, which we found quite confusing. We further then saw that for the PSIV iris, which is, if I quickly go back here, the PSIV iris is the strongest of the 3 irises. We basically have no effect on the no structure dependent effect on the translation initiation strengths, except that when you're knocking or disrupting the structural elements in the pseudoknot, it does actually increases the translation initiation strength slightly. Similarly to what we have seen in CRPV and PSIV, knocking out the different pseudoknots for the IRPV iris basically increases slightly the translation strength, and again the overall knockout of all 3 structural elements improves translation initiation. With that, we just realized that probably this is not that the structure of the iris is not driving translation initiation here, because there is no clear correlation between the presence of the structure or the maintenance of a structure in that element and the initiation strength. So now we were wondering, like, what actually does drive translation here? So we went into details and see how the different knockouts kind of like contribute to the effect. So we had a closer. Actually, what we then wanted to know is that basically how does that maybe relate to the growth phase of the cell? Because we are doing this here on a particular in the stationary phase, maybe the effects are different in different growth phases. So we picked basically one of the virus irises and looked at the expression level of the reporter protein, for instance, for that pseudonot2 knockout. And we can see here that over time in the growth curve of E. coli, the knockout indeed accumulates way more protein than the wild type does. And when this already occurs right in the linear phase, early, where the production of the reporter protein is higher than or faster, I guess, than in the knockout case than for the wild type. So we wonder, is this a initiation thing, or would that maybe have to do with maybe because we're liberating or removing a structural element from the mRNA, we are basically bringing down the mRNA stability, or we're increasing mRNA stability, or we have more mRNA that can be translated. So we then look basically at the mRNA levels quantitatively, and we can see that indeed liberating that structural element and decreasing the structural compactiveness of that translation of the iris decreases the mRNA stability, which that means taken into account that we actually have less mRNA, even also in the initial phase of that signal. That shows that basically the translation initiation strength is even higher, because we actually, although we have less mRNA, we actually make more protein. So yes, the interference with the structural elements does interfere with the mRNA stability, but it further shows that basically removing the structural element from pseudonot2 increases translation initiation strength. So maybe the problem was that these iris elements have evolved as an intergenic element. Maybe using it in the 5' end to drive translation is not providing them with the right context. So in order to test that, we then basically checked a dual reporter system where we have MRFP driven by a medium RBS, followed by the iris initiation element, and then the SGFP readout. So what you can see is that it does not make any difference if you're using a monocistronic element that we have seen before, or the bicistronic reporter construct. In either case, the translation initiation strength is not affected by the removal or the interruption of the respective structural elements here. So which means it's not context specific. So now we know that it's not context specific, and it is not a structure-based thing. So now we were wondering, what is the if this effect also occurs if you're using a reconstituted purified system where we have control. So, and if it occurs in that system, then the factor that gives rise to that increased translation initiation strengths of these disrupted systems, or if in a purified system, the structure-based translation initiation strength remains because we are lacking the complexing surroundings of the cell there. So we basically did the same experiment now. Instead of running it in E. coli, we ran it in a reconstituted purified system using our in-house compounds, and which basically is the all his-tech proteins from the TXDL system that we've independently purified the compounds from total tRNA from E. coli and amino acids and energy, and separately purified ribosomes from MRE600 that we purify in-house. And we can show that basically the effect is the same. No matter if you're in the cellular context or if you are in the reconstituted in vitro system, that pseudonot2 knockout has actually an even more pronounced effect in terms of driving translation better than the wild type iris element here, which basically means whatever gives rise to that increased translation initiation strength is not part of the complexity of unknown factors within the cell or the cell lysates, but it's part of the 54 components that we basically added to the system. So with that, we were wondering what component could this be, because most of the factors we actually do know what they do and what the purpose of them is in there. But we also noticed that when disrupting the structural elements, we're basically creating free AU-rich kind of elements that are not sequestered by a structural element, which might be an interesting binding site for ribosomal protein S1, which is part of delivering mRNAs to the ribosome. So we speculated or hypothesized that maybe ribosomal protein S1 is important for recruiting those destructured, I would say the wrong word, translation initiation elements to the ribosome. So what we did then is we ran the same reaction again, but this time we replaced the ribosomes with ribosomes from which we have removed ribosomal protein S1 to see if the translation strength has an effect, if removal of S1 has an effect to this. This also allowed us then to supplement back in recombinantly purified his-stacked S1 to the reaction and see if the effect can be reconstituted with that S1 thing. So that's exactly what we have seen. So in the case of the pseudonode-1 knockout, when we are using ribosomal subunits that have S1 removed, we basically completely abolish the production of our reporter protein, and when we bring back in here in checkered recombinant purified S1 into the reaction, we bring back translation initiation of the system. So we did not break our ribosomes, because those are the same ribosomes in these assays that went through the process of removing S1. And the effect is not specific to pseudonode-1 alone. It also occurs that, it's consistent with the essentiality of S1 for E. coli. Bringing down S1 down brings also wild type translation initiation down, and we can reconstitute translation initiation by bringing it in trans. So which indicates that allows us now, and I have not shown all the data that give rise to that model, but you can have a look at Luke's NAR paper for that regard. But it allows us to build a molecular model or mechanism in which the role of RRPS1 is basically the unwinding of highly structured 5' UTRs, so that a shined organo-like element in the start codon can be exposed, which is present in the viral iris that we're using here. And I actually can show this. I just had this on the previous slide here. Is when we are knocking out this shined organo-like system here sequence in our reporter construct, our translation initiation goes away. So it basically allows us to formulate a case where ribosomal protein 1 binds to structured mRNA, unwinds the structure element, exposes the start codon and the shined organo sequence, and then and then that will then allow translation initiation on the ribosome to occur in a the ribosomal binding site modulates the translation efficiency. Then basically it runs by that RBS, and that got exposed, and we have normal start codon selection and progression into elongation. And this can happen. This unwinding activity and binding activity of ribosomal protein S1 can occur on the ribosome and free in solution, so that either the RPS1 bound to the ribosome can bind the iris, unwind it, expose the canonical translation initiation elements and the related translation initiation site, or this happens on the ribosome, and that bound complex can then bind to the ribosome and start translation initiation. And this is of utility, and we have just shown that recently in an ACS-Synbio paper, where we could demonstrate that the addition of exogenous recombinant ribosomal protein S1 to the translation reactions, reconstituted translation reactions in the pure system, and this is now done with the pure system, not with our in-house system. The only thing that is in-house in this case is the ribosomes that have S1 replaced and the S1 that we have purified and isolated from our system. So you can see here that this effect, so this is corresponding to the, relatively corresponding to the effect that I've shown you before. Depending on what fluorescence protein you're using, you can improve the yield of that protein by increasing the concentration of ribosomal protein S1. And you see that that we are increasing the ribosomal protein concentration of S1 to 2.4 micromolar, which is in there. It's not too much, and it's related to the concentration of the ribosomes in the cell in that reaction. So it works for YFP, GFP, MRFP. Astonishingly, we didn't see any effect for CFP, but probably has to do with that particular sequence. I don't know. But what we also shown is that this not only works for the PSIV iris as I've shown you before, but also improves the translation initiation strengths of different ribosomal binding sites. As you can see, probably if I would have to do a ranking here, the effect is the strongest for a weaker ribosomal binding site than for a stronger ribosomal binding site. But in either cases, it improves this. We also have shown that it improves the translation initiation of all sorts of highly structured 5' UTRs in this reaction, which then basically means that being aware, for understanding the hidden role of ribosomal protein S1 as a robustness factor for overcoming structure in the 5' UTR is a critical design feature for these ex vivo translation expression platforms, I would say. So with that being said, the secret role or the hidden complexity that I was pointing at is the secret role of S1, which is not part of that general scheme, but in order to follow this scheme, there has to be the integration of ribosomal protein S1 in the overall reaction. So quick detour on other complexities that I mentioned before, and I will not go into details because we already have spent quite a time on the ribosome S1 story. But what we're also doing in the lab is we're looking at highly conserved translational or highly conserved GTPases that are present all organisms, and similar to the elongation factors that they're canonical known, like IF2, IF2U, IF1 responding, or EFG, or the ones that are responsible for secretion, like AFFH and FGSY. There are three, YIHA, HFLX, and YCHF, who are also conserved, equally conserved, but whose cellular function is not understood yet. So our lab has spent quite some time over the recent years to elucidate their function and tie them to the translation machinery, and all three of them are interacting with the ribosome. YIHA actually is a ribosome biogenesis factor, but HFLX and YCHF are involved in the function of the ribosome, regulating or mitigating problems that can occur during translation initiation. And so what we're doing in the lab right now is to study their functional role and to understand the molecular basis of their interaction. So we have made and so this is the graduate students that are currently working on this project, besides Dr. Brandon, who recently graduated from the lab, and the processes that we think those proteins are involved with mitigating this. With that being said, so quick introduction to the lab. There's, as I pointed out, there's correspondence to the different research streams by the different subgroups that we have, and this is the site in Lethbridge Prairie during pandemic and a wildfire, which is topical, I guess, right now with the Alberta forest fires. That's why it's so hazy there. We moved then basically east to the University of Manitoba, and unearthed, and all of you kind of that ever moved the lab understand that this is what the state is. And then when you run your first successful kinetics experiment, or whatever your pet peeve is you're very excited that you're ready and established and ready to go, which happened to us about a year ago, that we were able to restart doing kinetics with that. So there's a couple of students that in the lab during the last year and after the move have graduated. You have seen their publications, and that's the University of Manitoba campus. Maybe it will be in like a month or so because we're still brown. The snow is just melted. And with that, I'll leave it to questions.
Thank you so much. There's a lot of questions in chat. Can you see chat?
Oh, I ignored it for a second. All right. Okay.
If you could read the question out loud before answering, that's going to be great for the recording.
So yeah, so I'll just go through the questions from the top to the bottom and then see what happens. So I have a question that's asked if the algorithms that we're using also take into account the secondary structure of the mRNA. Or is this just simply a calculation between the interaction of the mRNA with the ribosome? So the Salus calculator has a couple of elements that they're using. They're using, of course, the predicted mRNA interaction with the ribosome, but also structural elements. So there you basically look at the delta G of base pairing. So yes, we also look into basically the structural, like some sort of secondary structure there. That's part of it. That's why you always when you're optimizing expression strengths, you typically you optimize your expression strengths based on the mRNA gene, or like look at the sequence that's behind your initiation element as well, because they actually contribute to the overall initiation strength. So you have to make sure that is. So that's why we're using the same message, so that we don't have that effect, so we can compare between them. I hope that answers that. Does molecular crowding affect iris binding? The cytoplasm is not aqueous environment. It's more like a technique. Yeah. So do you think we should be accounting for it in ribosome engineering, or the crowding interactions don't really affect the binding? That's a fantastic question. And the crowding element is, how do I answer this? So we have not checked the effect of crowding on this particular effect here, okay? Because for the assumption that we're doing in like how we ran the in vitro systems, that we wanted to run them in the aqueous system, right? But I agreed fully that that's a design feature that we need to have. What we did though is we increased, we added different concentrations of BSA to, which is kind of it's not a crowd that I would be using, but we check different BSA concentration on the effects. And there is only a limited effect of BSA on the translation initiation strengths, and that's in that Sheehan paper. That's in the supplemental to that where we tried different BSA concentrations to that. But it's an interesting factor to do. We did a systematic study of crowding on nucleotide binding properties of translation factors and tRNA delivery onto the ribosome with all sorts of different kind of polyglycol kind of crowder agents to this. The effect has not been so strong that we would have pursued it further, I guess is my answer to that. Hope that answers it. So I have a question from Agnes here. So do you see differences in expression if you put the same iris in front of different types of proteins? For example, use 3 or 4 different reporter proteins of different iris sequence. Would the strong iris always be strong, or would it be dependent on it? So we have done this with the different fluorescence reporters, and the strong iris is always a strong iris, and the weak iris is always a weak iris, if that answers your question. And you probably have seen that before. Brian asked, do results from cell assays translate well into the cell-free system? Did you try those iris elements in cell-free? So we have not done, so the in vitro systems that I've shown you is basically the pure system or our in-house purified reconstituted system. So this is, and for that pseudo 2 knockout, they're basically indeed comparable. But actually what the nice thing is, because you might remember that the accumulation of GFP was stronger in the in vitro system than it was in the cellular based system. The benefit here is we're basically we have no RNAs present that come from the cellular system, or So which means the lifetime of our mRNAs is higher actually in the cell, in the in vitro reconstituted system. But basically the iris elements that we tried in vitro and in vivo, they translate very well. Did you control for transcription efficiencies? Yes, we did measure the mRNA levels, and we have seen different. So the different irises do affect the mRNA stability, and we have shown that for that one case. And within the different irises, we also see if you interfere with the iris structure, you see changes in the mRNA stability. But we can map them to the protein expression rate, and actually, as a matter of fact, you can also map the expression strength to the folding energy of that iris element. So you can do a correlation between this, like how structured, like how tight the structure is, and the expression strength. So which means the more, like the more structured and the more energy, so like the more favorable the structure is, the less expression you get of the protein. So in the absence of, well, yeah, then requires wise the ribosome protein S1. Ribosome question, what evolutionary advantage? Okay, general ribosome question, I like that phrase already right there. What evolutionary advantage does the complex ribosome initiation process with its many parts provide? What is the problem with a ribosome that does not disassemble completely after termination? Okay, excellent question. So this falls into two questions. Why is translation initiation so complicated? Well, you know, the classic answer that I always give in class or textbook is like regulation, because then you can regulate the expression strength. You have different multiple committing steps. Yeah, so it's a regulatory thing. It also allows a design. It's a design layer or regular. Yeah, well, it's basically regulation. The design layer and what kind of mRNAs are going to be more likely to be translated, because it overcomes. It also correlates with the availability of the different mRNAs. Also, you're using the same codon to initiate on. Then you actually have a message codon. You need to find that right spot, right? What happens to a ribosome? What ribosomes? There is initiation on non-disassembled ribosomes does occur, but it's a non-standard process. So you can phase in an mRNA on a tight 70S ribosomal couple, and then basically bind initiate a tRNA and jump into translation initiation. Well, into the fully assembled translation elongation competent initiation complex without going through the initiation step, but it's non-canonical. Okay, so I missed, where did I go? It moved. We did this, we did this. Do you control for transcription? If we did this, right? It is great. Do you think iris elements would work similar on tethered ribosome? Yeah, interesting question. Well, you know, the funny thing is, it requires, well, they will bind to this, right? And I think it will depend on the availability of ribosome S1, right? So which means they should totally work on those tethered ribosomes if you give them enough S1, I guess, right? And then you have the full gamut available. Potentially ridiculous question, I do not know that. What prevents recreation of a bacterial initiation system RNA based using eukaryotic ribosomes? Bacterial initiation system. I'm not quite sure if I understand that question properly. So the translation initiation in eukaryotes is different because it actually uses the kind of thing, or the factors bind to a different site. The whole kind of like identifying the start codon and identification of the mRNA to bound to is completely different, right? If you're asking the question, if you can engineer a eukaryotic ribosome in a way that it would like do Shine-Organo-based kind of translation initiation and vectors. The question then is why would you do that? Because then you can basically use the bacterial system, I would think. But I guess your question goes into exactly in what we were intrigued with initially when we saw the binding of the iris element to the translation element, right? So, you know, to the ribosome, because maybe you can swap that. Okay. Okay, Maya, that makes sense. So have you titrated YCHF, HFX, etc. into pure system to see if it improves reduces protein output, blah, blah, blah. Yeah, actually, so we have done. Not all of them do, because I think we do not 100% understand at what stage they're going to be beneficial. Okay, so I can tell you, for instance, if you titrate in too much of HFLX, you're going to kill the system. Okay, and that has to do with the fact that HFLX is a ribosome splitting factor that we have shown that a couple of years ago. So which means if you're increasing the concentration of HFLX, you're basically counteracting initiation. So you have to be careful with that. HFLX has to be there for a particular case. For instance, if you're running translation of whatever, like if transcription is not complete and you have an incomplete mRNA, ribosome initiates on that incomplete mRNA, runs towards the end of the mRNA, and then stalls on that mRNA. Then HFLX can come in and disassemble that ribosome and relieves basically the stall and anything that's behind that ribosome, and these ribosomes then can reinitiate. Okay, so you need to find the right concentration for that. That seems to be a system, you know, you can play then, like, you know, if you know how efficient your transcription is and how many incomplete transcripts you have, that will then have an influence on how much you're going to have to put into the HFLX component to this. But exactly, that's the point. Yes, that allows us, and that's the new thing what I was saying, you know, this hidden complexity of stress response in the cell allows us to fine-tune these reconstituted systems. I think that's it, is it? Did I answer everything?
Yeah, I think you went.
Lab A does, if you want to know.
Okay.
Yeah. Lab A does.
I think you went through all the questions. Yes. I was really curious to hear your answer to the tethered ribosome question, because that was something that I would be interested in too, especially if you modify ribosomal proteins more.
Yeah.
All right, if there are no more questions, I want to say thank you so much again for the talk, and thanks everyone for the discussion. And have a great rest of your week, and see you all next week.
Okay, thank you. Bye-bye.